Good afternoon, everybody. Thanks so much for joining us today. My name is Bruce Holst. I'm the Vice President for Botany at Selby Gardens here in Sarasota, Florida. And welcome to a very special virtual botanical presentation. This is in collaboration with Lawrenceville School in New Jersey, Glenville State College in West Virginia, and the National University of Columbia in Bogota, Colombia. This last month has been a real interesting experience for us. We have had 8 guests here for the past month studying the molecular systematics of the Gesneriaceae plant family, one of our favorite plant families with a large percentage of epiphytic species. The team is led by Dr. John O. Clark, Aldo Leopold Distinguished Teaching Chair at Lawrenceville. John has had a distinguished career at the Smithsonian Institute and the University of Alabama. He has conducted field work throughout tropical America and is a great friend of Selby Gardens, having donated thousands of herbarium specimens to our collection. He's also a research associate of ours and has been collaborating with us since the 1990s. So I'm going to turn over the rest of the presentation to John, and I think you'll find the work that he and his colleagues and the students have been doing is nothing short of spectacular. John?
All right, hey everyone, it's good to be here. I think you can see my screen. So great, just an overview of what's going to happen here. I will provide some brief comments, and then my colleagues will also share some comments. We'll have 2 15-minute presentations, and the way it's going to work is send me comments in the chat, and so we'll have questions at the end. Okay, can you all hear me? Is it good? All right, so you can put questions in the chat, and then what I'll do is I'll hold on to those questions, and at the end of the 2 student presentations, we'll get to those. But as the students are presenting, just send us questions. I want to thank in particular the parents of these 4 students, because given the context of COVID, this trip was much more complicated than our typical programs at the Lawrenceville School. These students are selected, they're rising seniors, they're selected from a large pool of applicants, and they are our most prominent science students at the school. At the Lawrenceville School, they're in the Hutchins program, and Dr. Elizabeth Fox and Marquis Scott played a big role in getting this program running, and we're all really grateful to them. I want to thank the Selby Botanical Gardens, and in particular, I want to thank all the wonderful volunteers who have welcomed us and made us feel at home here, and been very kind to us, have worked with our specimens, have helped us locate images that we needed for our research. And so I want to just give a big shout out to them. I want to thank Wendy Deming and Jennifer Romanecki for transforming this place. Wow, it's amazing since I came here in the 1990s to see all the great changes. And so thank you to them for hosting us and making this experience possible. I want to thank the scientific staff, Dr. Sally Chambers, Liz Gandy, Sean McCourt. Thank you for also hosting us and making our stay pleasurable and informative, and working with us every single day. And in the Sarasota area, there's a few people I just want to mention. You'll see a lot of pictures in the presentations today. And so if you want to know where I learned to take pictures, I learned from Rick Dunn, who I had a nice dinner with the other week. Rick, when I was 22 years old, he brought down to Ecuador a digital camera, taught me how to use it, left it with me. And so a lot of the pictures you're seeing today are a result of Rick's help in guiding me when I was a young 20-year-old as a Peace Corps volunteer in Ecuador. Nancy Cast, she's been growing lots of really important plants. She's a meticulous record keeper, so we know very well, where the plants came from, how they're distributed, and she's been helping me and the growing community, the horticultural community, for more than 2 decades. And so just very, very grateful for her horticultural skills and sharing that with all of us. I want to thank Jean Katzenstein and also the Gesneriaceae Society. Financial support for this project came partly from the Gesneriaceae Society. And Jean, who's been a huge advocate of the family, mentoring students and researchers for her whole adult life. So thank you very much. It's been wonderful to see Jean here in Sarasota. And then I also want to thank the collaborators. This project was possible because we brought together a team of scientists. Dr. Clavijo traveled from Colombia. Dr. Jeremy Keene traveled from West Virginia, and he brought with him Tori Portfeld, who is his undergraduate honors student, and she worked very closely with the students every single day. So I want to thank them for their collaboration and their help. And they will also speak after I'm done and share a few brief comments. And then of course, Bruce, thank you. Bruce and I have been talking about this for over a year, and he made everything happen. So we're all just so grateful to Bruce for not just hosting us, but also showing us around town, showing us to some natural areas, sharing his botanical knowledge. And so just all around, thank you very much. Now, before the students present, I just thought I'd present two just broader pictures about the plants. The plants that they're sharing today, members of the flowering plant family Gesneriaceae, they come from critically endangered areas. So if you look on the left, this is a map of Ecuador. It's what Ecuador would have looked like vegetated in the 1950s. And then if you look on the right, you'll see a vegetated Ecuador in 1996 based on satellite images. And there's just especially west of the Andes, there's very little rainforest left in those areas. These plants came from difficult to get to areas. So this is an easy access road. You can get a car in and out. Here's one of my students walking on a muddy road. This sort of shows you where these plants came from. So there's a huge amount of effort to get them here. And the students participated in these expeditions. But unfortunately, when I go back to school in the fall, we have 820 students at Lawrenceville, and only 130 will have experienced a full year of in-person classes. There's only 2 students on campus who have actually experienced a field expedition, and one of them is one of the presenters today. And so Selby allowing us to come here and do some research, it's not only been important for their education, it's allowed us to do something in person, and we haven't been able to do that in over a year. Just think about that. So thank you. Thank you very much for providing that opportunity to us, the researchers, and our students. And the other thing about Selby Botanical Gardens is that there's an incredible live collection, there is an incredible herbarium, and there's a molecular lab facility. And we were able to use all 3 of those together to develop these projects that you'll hear about. So thank you, Selby, for the vision and for providing those resources and for providing that to us. The other thing I just mentioned is that the students are talking about how we classify plants based on evolutionary relationships. So if you look on the left, there's a lot of circles. And I think in the past, we've had this idea that, okay, if Columnia has a berry, then everything that has a berry has to be Columnia. And so you sort of fit things in the circles with the characters that you understand. And that's sort of one way, and that's the traditional way that we used to classify biodiversity. Things have a character, they fit in the circle, and you figure out that circle where they fit. But what happens if a Columnia has a capsule? Like in the lower left here, I have a Columnia with a capsule. Well, that makes it more complicated. And so what we're doing is we're providing an evolutionary framework for understanding the plants that we study. So if you think about it, not in a plane, but as an evolutionary tree, then the things with berries, they're basal to everything else. The things with capsules in Columnia are basal to everything else that have berries. So here's another way to look at that. So if capsules evolved and then berries evolved from capsules, if you think about that in an evolutionary framework, sure, Columnia can have berries and capsules. You just have to understand it from the phylogenetic or the evolutionary context. So 1, we promote biodiversity. 2, we want to understand that biodiversity in an evolutionary context. And then that is sort of how we wanted these student projects to work. So hopefully we'll have a lot of questions. And what I'll do now is I will stop speaking. You'll see me at the end of the presentations, and I will turn it over to Dr. Laura Clavijo. Thank you very much.
Hello, everyone. So I am Laura Clavijo. I am an assistant professor at the National University in Colombia and a curator at the National Herbarium of Colombia. My main focus in Gesneriaceae is the diversity of Gesneriaceae in my home country, Colombia. I've worked for many years in the taxonomy and evolution of the genus Drimonia, which you can see several images at the bottom of the slide representing the species of the genus Drimonia. And in the past 3 years, I've been working on the taxonomy of the genus Clariho and all the potential hybridization that happens within the genus. All these research that I do, I've been able to do it thanks to the support of many people and many institutions, and one of them has been Selby Gardens. I was an intern here back in 2008. It was an internship funded and supported by the Gardens and also by the Gesneriaceae Society. During that time, back in 2008, Bruce and Jean were the people that were always with me and helping me. I was getting ready to get into my PhD program under the supervision of Dr. Clark. I was lucky enough to get in and complete it. So I just want to say thank you to Selby Gardens for always opening the doors for me, to Jean and the Gesneriaceae Society to support my research and now my students' research, and of course to John for organizing this program, leading the research here, and continue collaborating with me. We are here in this image holding a new species of Drimonia with the largest leaves. So it has been a very productive collaboration, and I'm really happy that I was able to come here and contribute to these two really interesting projects that you are going to hear from the students. So thank you, everyone. And then I'm just going to pass it to Tori, I think. And thank you for coming.
Hello, everyone. I'm Victoria. Many of you know me as Tori. I am a Glenville State honors student from Glenville State College in West Virginia. I am conducting my undergrad research here with Dr. Jeremy Keene, and I have been conducting research with him for almost 2 years now. I have been helping him with the taxonomic relationships and delineating species within the genus Diastema. I would also like to just say thank you to the Gardens and everyone who helped collaborate with us and allowed us, not just me, but the other students as well, for this opportunity and this unforgettable experience. It's been amazing and I have learned so much, and I will turn it over to Dr. Keene.
Hello, everyone. Many of you probably already know me either from the society or from previous presentations. I am an associate professor of biology at Glenville State College. And Selby was, or my first presentation was actually at Selby Gardens. I came here the fall following the start of my dissertation research. I came to the World Gesneriaceae Conference and presented my poster towards the revision of Diastema and Monopyle. And I was able to work through all of the collections here while I was visiting, and it really jump-started my career overall. Over the next decade, because it's been about a decade now, I have completed my dissertation, of course, and I'm able to give names to a lot of these things now. With all of the wonderful pictures that John and Lara and others have taken, I can see what some of these things look like a little bit better in the field as opposed to on a piece of paper, and some of my own pictures as well, but John takes wonderful pictures. So I've got several here on the side and a picture of me here at the bottom at the herbarium 10 years ago. But we really returned here to look at the specimens over again and look at them in context of additional sampling that the girls were, the ladies were able to do. And they've done a really great job, and I hope you enjoy these presentations. And I would like to say thank you to Selby Gardens, of course, Jean Katzenstein for always being there, always helping me out with either my students or otherwise, and of course the Gesneriaceae Society for all of their support as well. Okay, and I will turn it over to Nikita and Nicole.
Thank you so much, Dr. Keen.
You're welcome.
Good afternoon, everyone. My name is Nicole Cheng,
I'm Nikita Kafasetti.
and today we're going to be talking about the final frontier of exploring relationships and generic boundaries of New World Gesneriaceae, a molecular phylogeny of diastema and monophyly. During our month-long study at the Selby Botanical Gardens, we were working with Gesneriaceae. The way that you can tell Gesneriaceae apart from other families is their paired flowered sign, where the terminal flower opens first and the front flower opens second, which you can see in this middle photo. In addition, Gesneriaceae contain opposite leaves, meaning leaves opposing each other on the stem, which you can also see in this Weber photo. The anthers, which are the parts of the flower that produces pollen, are coherent, meaning that they're set together but not actually connected. You can picture it as two hands together in prayer. The bottom right photo shows a unilocular ovary with parietal placentae. The lining cells make up the walls of the ovary, while the white spaces are actually connected and make up the one locule. The parietal placentae are parts of the walls that dips inwards, and the seeds are the parts that kind of look like eggs within the ovaries. To give some background information, there are currently 22 accepted species of monophyly and 20 accepted species of diastema. The species diastema and monophyly ranges from Mexico to Bolivia and parts of Brazil. Colombia, Peru, and Ecuador are likely the most diverse places for diastema and monophyly because of the humid and wet climate along the eastern Andes. Recently, in 2013, Alain Chatems et al. Discovered unidentified populations of diastema in northwestern states of Brazil. Until then, no one had known that diastema occurred in Brazil. There was evidence from previous phylogenies that diastema was polyphyletic. So as one of our objectives, we wanted to compare diastema to another genera within Gloxinianae. There was also evidence from previous phylogenies that monophyly was monophyletic. So we wanted to confirm this with more taxa. Additionally, we reevaluated the subtribe Gloxinianae with increased taxon sampling to show the generic relationships in the subtribes. Most other studies don't have more than 50 samples, and the most diastema in a study have been 3 or 4. So the 150 samples in our study is a very remarkable increase, especially because all the samples we used were diastema or monophyly.
And now for some background information. This is a morphological and molecular phylogenetic tree from the paper by Rolson et al. In 2005. We see here that monophyly is monophyletic, and the traditional diastema clade and diastema vexans clade are unrelated. So diastema is polyphyletic. We anticipated our sequenced diastema to fall either within the traditional diastema clade or with diastema vexans. The reason why we're comparing diastema and monophyly to the subtribe Gloxinianae is because different topologies have been seen in previous phylogenetic trees that have been produced. And to give some visual context, here are images of the different diastemas. C is diastema commiferum, A and E are diastema racemiferum, and G is diastema vexans. You can see that diastema racemiferum and vexans look similar, but it's not because they share a recent common ancestor. It's just an example of convergent evolution. Monophyly is, again, monophyletic in this tree, so we didn't put pictures of it. We would anticipate that they would all look pretty similar to each other. These are some photos of diastema vexans. One of the exciting things about working at Selby was that we could work in the lab and then walk into the herbarium right next door to see the specimens that we were sequencing. And we discovered that diastema vexans has recently been collected in the field and not only in cultivation, which is something that we didn't know before. One of the 100 samples that we sequenced actually represents a second specimen that was not from GenBank, but actually from recently collected material. And here we are looking at specimens with Tori and Dr. Keen. This is another molecular phylogenetic tree from a paper by Clark et al. In 2011. It shows that monophyly is monophyletic again, and it shows similar relationships to what we saw before with diastema, vexins, and traditional diastema. This paper doesn't have significantly more sampling in diastema or monophyly since Rolson in 2005, so there isn't much difference between the trees. So it was especially exciting to add more than 100 samples to this phylogeny. And now we can get into our own research. We used samples from papers by Araujo et al., Clark et al., and Rolson et al., so that we could compare previous data to our own outcomes. There were 149 total samples, and about 75 haven't been represented in GenBank and came from our original sequences. We used Rhizophyllum and Gesneriaceae as outgroups because this subtribe, Gesneriaceae, is most closely related to Vaccinianae. Although we did collect data from TRNH, PSVA, GFlow, and TRNTL, we ultimately chose the internal transcribe spacer, which is ITS, as a marker in the trees. After we had our sequence data, we used maximum likelihood analyses performed in RAXML with 1,000 bootstrap replications for our support. Here's a picture of our workstation and the materials that we use. The pipettes allowed us to work with really small amounts of DNA and chemical reagents. We use a thermocycler in the lab to make many copies of our target DNA using polymerase chain reaction, or PCR. And then we ran gel electrophoresis and used the gel imager in the bottom left to visualize our DNA and make sure the correct band lengths of ITS corresponded to the ladder DNA. And here's an image of what we produced with gel electrophoresis. The ladder DNA all the way on the left is used to confirm the size of the gene region that we amplified. And all of our samples are similar lengths that correspond to the ITS region. In the last well, we have monophyly puberella, which will show up on a tree later. We use Genius, which is a bioinformatics software, to align our DNA and produce a simplified phylogeny to check our data. And now onto our results. This is a maximum likelihood tree with 1,000 bootstrap replicates for support. We also ran a maximum parsimony analysis that was congruent in topology to the tree that we're presenting right here. This tree shows a similar topology to what was retrieved by Rolson et al. And with our expanded sampling, we see 5 diastema-like clades marked in purple. We also see 2 monophyly clades in blue, which we will come back to later. This is a subset of our data detailing monophyly. This phylogeny shows us a sample of species that have been in cultivation and what you would traditionally think of as monophyly. As for the characters, monophyly retains anisophyly when there is one big leaf and one small leaf that oppose each other on a stem. All of the samples in monophyly have hooked trichomes or hairs, variable swelling in internodes, osmophores that produce the scent, and campanulate corollas, which means a bell-shaped flower. These are 2 undescribed species that we sequenced showing anisophyly, which is again one big leaf and one small leaf across from each other. Monophyly also has the variable swelling. You can see that between the nodes, whereas in other genera, the swelling would occur at the node of the leaf. This is a new species of monophyly from Colombia. It's a good example of the campanulate corollas, which means the bell-shaped flower. And only about 5% of monophyly have these nice yellow flowers, so it was really great to see some photos of it. This is monophyly sodirawana, which was only known from the type collection, and it was destroyed in Berlin, in the Berlin herbarium during World War II, and that caused a lot of confusion about its identity. So this shows a really nice picture of it, one of the largest flowers in its genus. And monophyly ecuadorensis is very floriferous, which means it has a lot of flowers. And until recently, this species has been confused with monophyly macrocarpa. And one of the things we noticed when going through the images is if we look in the flower, we see a lot of black flecks, as you can see on the left photo. The flowers attract arthropods, which are the flecks, often found near the osmophore at the base of the flower that produces the scent.
This is a subset of our data that shows a detailing of one of the diastema clades. One thing to note is that all the names we have on here are most commonly encountered in herbaria. It just shows that there is a lot of confusion in how diastemas are identified. Also, each of the clades here represent 10 or more samples, even though in this form it may just look like one. On one of the bottom groups, you can see diastema species axil. There are more than 5 species within this group, and they all have axillary inflorescences, meaning that all the flowers come out between the stem and the leaf in the axil, and they're all new species. This was a diastema that was seen in Ecuador, and this just shows an example of an axillary inflorescence. The 3 marked diastema species Aniso have anisophilous leaves at the apex, meaning that there is one small and one large leaf opposing each other. And they also have axillary inflorescences. We believe that with expanded sampling, that all the species with anisophilous leaves will come together with one clade. Each of these groups represent multiple samples, and like the samples within the diastema species axil group, they are all new species as well. This is just an example of a diastema with anisophily and also axillary inflorescences. The 1 marked diastema racemifera-like are the traditional diastemas. They have terminal inflorescences, meaning the flowers grow from the end of the stem, terminating the growth of that branch. This is an example of a terminal inflorescence in a diastema species. This is a diastema species that was photographed in Ecuador. It's an example of the scabrum-like lineages. This lineage is smooth and has no hairs. There is also still ongoing work on the morphology to match the scabrum-like species to clades. This is another subset of our data showing the vexins-like lineages. This lineage is very different from all the other diastema lineages. By these pictures, we can see that diastema vexins is another lineage that has converged or morphological similar to other diastema lineages since it shares some characteristics. And here is our last subset detailing diastema. We can see that Diastema fimbrata is sister to Chatemsia. With Diastema species nove, we see another lineage from Peru that is basal, which means the common ancestor gave rise to Diastema species nove, and it also gave rise to Diastema fimbrata and Chatemsia. The common ancestor has lobed leaves, and previously it was unknown. And the bottom left photo shows a picture of the Chatemsia fruit. Monophyly reflexa is also here in this subset, which would make monophyly polyphyletic, meaning to be in multiple areas of the tree. But the species doesn't fit well with the concept of monophyly. Monophyly reflexa was simply put into monophyly because there was no other place for the taxon at the time. So we can still say monophyly is monophyletic. This is an example of the Diastema species nove I was talking about earlier. And here are some more examples of the species I was talking about. The top right and bottom left photos are of a Diastema fimbrata from Peru, and the top left photo is of a Chatemsia from Brazil. In monophyly, the flowers morphologically are very different from each other, but the monophyly reflexa flower is especially different from what we would imagine a monophyly to look like. Again, this just shows how monophyly reflexa does not fit well into the monophyly clade. To summarize, the conclusions we have drawn from this study is that Diastema is polyphyletic and is currently separated into 5 evolutionary units across the Gloxinianae subtribe. In addition, monophyly is monophyletic, and our phylogeny shows that monophyly reflexa belongs to a different lineage. Finally, in the future, we will look to collect, sequence, and describe the mystery Gesneriaceae on Flickr. It will be interesting to see if this is a diastema or not, because diastema is artificially defined and we do not know a lot about it. Our phylogeny will be compared with different specimens to understand morphological differences. Additional sampling near the diastema and Niso group allow for better refinement of clades. Lastly, we would like to thank all the following people for their help. We are especially grateful for the support and guidance of Dr. Clariho, Dr. Keene, and Dr. Clark in this project. Thank you all so much for listening.
Thank you, Nicole and Nikita. Hi, everyone. I'm Brian Fan.
I'm Ben.
Here's our presentation. Taxonomic names are not set in stone. A phylogenetic approach for tattoos and generic names of Central American Gesneriaceae.
On the left here is a photo of Kelvin Cooper, who lives in the Sarasota area. He has a very unique tattoo of a plant called Alsobia baroniae, and on the right we have an actual picture of it. It was taken at the live collections at Selby. I will be focusing on the Alsobia and Covenanthus genera, while my colleague Brian will focus on Orsidina and Rufidorsia. The objective of this study is to determine whether or not Alsobia is monophyletic, and to determine the phylogenetic relationship between the Alsobia and Covenanthus genera. Are they one genus or two genera? We know that Alsobia, Rufidorsia, Orsidina, and Covenanthus are strongly supported as a monophyletic group from previous phylogenetic studies. The first study is from Clark et al. 2006 in Taxon. A 98 bootstrap value, as you can see in the red circle, represents a strongly supported node, and we can see that the 4 genera under that bootstrap value make a monophyletic group. But what does it mean to be monophyletic? In phylogenies, monophyly refers to the condition of being a clade. A monophyletic group includes all descendants of a recent common ancestor. As you can see, part A of the diagram to the right is a monophyletic group, while the others are not. If you wanted to classify bats and birds together based on the evolution of flight, they would not be monophyletic because they didn't evolve from the same recent common ancestor. Flight would be an example of convergent evolution. So returning to the background studies, the second study was Rolson and Roberts from 2017 in the Systematic Biology Journal. It again shows that the 4 genera are a monophyletic group, and it is strongly supported by a 100 bootstrap value. The third and final study is Serrano et al. 2017 from the Proceedings of the Royal Society Journal. Once again, this study evaluates the 4 genera to be monophyletic. So we aren't going to show the results for the 130 species in this study because they are already supported. This allows us to shift the focus to the relationships among the 4 genera. In this study, we are looking at the Gesneriaceae tribe and the Columniae subtribe. We included 130 species as shown by these beautiful images in our analysis for ITS. In this lab, we generated ITS data for 26 species, and the rest of the data came from GenBank and from Clark et al. 2006, which is a paper published in Taxon. For the materials of the study, we used extracted DNA samples from tissue. We also used 13 plant samples generated by Selby. Lastly, we used sequence data downloaded from previous studies on GenBank. A lot of the species whose DNA we worked with and sampled are found at the greenhouses in Selby Botanical Gardens, and many original samples came from 2 field expeditions to Panama. On the right here is a picture of us at our workstation, and we are visualizing a PCR product. Here are some pictures. On the left, we can see Dr. Clark on a field expedition in Panama. In the middle and right pictures were taken in the Selby greenhouses. In the middle picture, you can see Angel Lara, VP of Horticulture at Selby. We took the DNA samples and amplified them using PCR, or polymerase chain reaction. We visited our products using gel electrophoresis and then sent those products to be sequenced at GENIUS. We used Genius bioinformatics software for alignment and contig chromatograms and analysis. Lastly, we used ITS4 and ITS5 primers in our PCRs, which are usually around 20 bases long, and the region we aimed to PCR in our samples were over 750 base pairs long. Beware of GenBank sequences. Some GenBank submissions have lots of ambiguous base pairs. We went through the GenBank data and excluded some samples because they were messy sequences. In our alignment, an N denotes an unknown base pair, and lots of them can make sequences unreliable. Here we see that Rufidorsia major is nesting with Neomortonia rosea, and it is an incorrectly identified specimen. For that reason, we refrained from using it in our study. If it were a correctly identified sample, it would be nesting with the other Rufidorsias in the bigger blue region on the tree. So in order to determine the relationships between Alsobia and Covenanthus, we must look at their morphological features. For Covenanthus, the flower is actinomorphic and star-shaped with bright orange calyx. What's special about Covenanthus is that it's a monotypic genus, meaning that only one species has the Covenanthus genus, and that species is Covenanthus calyclamis. The fruit, which was recorded for the first time in 1968, is a loculicidal, fleshy, bivalve capsule with rather thin carpal walls. Its leaves are simple and opposite with an elliptic shape and serrate margins. Alsobia is strictly epiphytic, so it can only grow on other plants. There's typically one stolon per node in alternating leaf axils, and these stolons arise at a leafy node and terminate in a leafy node. Leaves are petiolate, blades are elliptic, and margins are variously toothed. Stems, flowers, and fruits of Alsobia have simple and multicellular hairs and glandular hairs present within corollas. Its corollas are typically oblique in calyx, white, and mottled with color. Now that we know the morphological characters that define each genus, let's compare their characteristics. The Alsobia flower, which is on the left, has 5 lobes that are white and mottled with blue or purple. On the other hand, the Covenanthus flower is a yellow and somewhat inflated tube. Here, the Alsobia fruit is a fleshy 2-valved ellipsoid with numerous brown seeds. In contrast, the Covenanthus fruit is papyraceous instead of fleshy and capsular with thin carpal walls. Hans Wheeler illustrated and described the fruit of Covenanthus, but we are not aware of any images that show the mature fruit. It may be that all the cultivated material available is likely the result of vegetative reproduction. We are not aware of a Covenanthus fruit in cultivation. We challenge you, Angel Lara, VP of Horticulture, to grow and photograph a mature Covenanthus fruit. On to our results. We sequenced Alsobia broniae and Alsobia chiapensis, which are not represented on GenBank. Alsobia was previously considered monophyletic, but with the inclusion of these new species in our maximum likelihood tree, Alsobia is rendered paraphyletic. Based on our sequences, Alsobia broniae, which is where the tattoo nests, and Alsobia chiapensis are more closely related to Covenanthus calyclamis than any other species under the Alsobia genus. In evolutionary biology, this is called paraphily. So what are the takeaways? Tattoos of plants are okay, but it's probably best to refrain from including binomials, as these names are subject to change. Follow Kelvin Cooper's example. Use the image, but avoid the scientific name. It is quite possible that the Covenanthus will become an Alsobia or vice versa. Future work. In the process of our research, a new species of Alsobia or Covenanthus was encountered and uploaded to iNaturalist by Jan Meerman. iNaturalist is an amazing platform used to document biodiversity, and we all have iNaturalist accounts. This new species of Alsobia was found in Belize by Jan, and future work will revolve around sequencing it and placing it in a phylogeny. We know that it's an Alsobia or Covenanthus because of its stolons and because of the shape of the corolla. This Alsobia was found in Pueblo Viejo, Belize, and the red dots represent other locations where it was documented. Bruce Holst is organizing an expedition to see if he can collect it later this year.
Thank you, Ben. For the next part of this study, I'll be focusing on Orsidina and Rufidorsia. We have 3 main objectives. The first objective is looking at whether or not Rufidorsia intermedia and congestive flora are conspecific, meaning they are the same species. The second objective is exploring if Orsidina and Rufidorsia are 1 or 2 genera. More specifically, should Orsidina be classified as a member of Rufidorsia, and if not, should Orsidina be recognized as its own genus? Lastly, we're looking at whether or not Rufidorsia and Orsidina are monophyletic. Ricardo Kreibel lumped Rufidorsia intermedia and congestive flora into 1 species and called both congestive flora in the journal above Manual de Plantas de Costa Rica. Hans Wheeler originally recognized 4 distinct species from a genus he described. From those 4 species, Wheeler recognized Gesneriaceae intermedia as 2 separate species in the Selbyana journal above. It was only the second issue of Selbyana in 1975. These are the 4 species of Rufidorsia Wheeler recognized. He described 3 and reclassified one as a species, which is the Rufidorsia congestiflora. He describes how different species of Rufidorsia were found at slightly different altitudes, and Wheeler proceeded to describe how these plants differed at various altitudes in terms of their leaf characteristics. The altitudinal ranges of Rufidorsia overlap significantly, as Wheeler recorded Rufidorsia intermedia in the 800 to 1,000 meter ranges, and the congestiflora in the 850 to 1,500 meter ranges. Furthermore, visually, the 2 species appear to be extremely similar. The leaves are similar with roughly the same type of hairs and a similarly shaped leaf. There are clearly significant morphological similarities based on these depictions. Wheeler described Rufidorsia as having a comparatively small white corolla. Dorsal side of the 2 upper corolla lobes are colored brick red with fruit in the form of white berry. There is bilateral symmetry, and Rufidorsia are epiphytic, meaning they can grow in other plants. The Rufidorsia also have a small 2-tone corolla, which is white and red, and the dorsal sides are brick red. The Rufidorsia major calyx has bilateral symmetry, united anther, and leaves appear to be very smooth. The Rufidorsia congestiflora has corolla lobes that are unequal. The leaves are long, dense, and the calyx lobes are equal. The seeds dry and have a reddish-brown color. The minor is the smallest and the major is the largest. The major and the minor share many morphological similarities, but the major is essentially a much larger version of the minor. Wheeler also recognized and described 2 species of Orsina, with the Orsina sericola and the Mexicana. The Mexicana is very unique, and we'll be talking about it later in our presentation. The fruit of the Orsina is pointed berry. The berry is translucent and appears black due to the seeds on the inside. It has erect or ascending stems and has opposite leaves of equal size. The corolla are a short, somewhat inflated tube. Orsina are epiphytic, perennial, and the calyx lobes are subequal. The flower is uniformly pink and suffused with some white. What happened to Orsina sericola? Wheeler published the genus Orsina sericola in 1977, which included 2 species. He named it after Danish botanist and explorer Anders Orsted. And so you can see in the middle, there's a photo of him. Here, the Orsina flower is uniformly reddish pink, but Rufidorsia is dark red on the top, but white with very small red specks in the bottom half of the flower. It looks like the Rufidorsia flower has a distinct red roof on top of the flower, which is very significant. The Rufidorsia flower also shows a sharp contrast between the colors of the flower in comparison to the Orsina flower. Here, the Orsina sericola fruit, as I mentioned before, appears to be black, but it's actually transparent. The seeds inside are black, so you can see the black seeds through the translucence of the fruit. This is not described in literature and is something that we found in the process of doing this project. On the other hand, the Rufidorsia fruit is white and non-translucent. Orsina also has a pointy berry, whereas Rufidorsia has a flat berry. The Rufidorsia fruit also has much more hair than the Orsina fruit. Orsina and Rufidorsia are sympatric, meaning that they are found in the same areas. The shapes represent various locations of different species of Rufidorsia in these areas. The Orsina significantly overlap in location, but there is one species of Orsina, Mexicana, which exists in an isolated environment. We included all the species from Panama and Costa Rica. We do not have one from Mexico, and we only know one collection of that species from Mexico in 1972. The one from Mexico is only known from a type locality and type collection. Here is a tree representing and showing the relationships between all the species we sampled. The 4 genera we focused on were Orsina, Rufidorsia, Selby, and Copenanthus. We will go through the process we use to simplify and condense our tree. We generated 13 of these samples ourselves through the use of PCR. The samples highlighted in yellow are the ones we generated. Many of these samples were taken directly from the live collection at Selby. The 13 other samples were from the 4 genera were taken from GenBank. Other genera were collapsed. Because our central focus lies in Orsina, Rufidorsia, Selby, and Copenanthus, we simplified our tree by collapsing species that lie outside of the genera we wanted. As my colleague mentioned, the red dots and the branches represent bootstrap values of 95% and over, meaning there is a high confidence level in that clade of the phylogenetic tree. Here is a summary of the specific relationships we're looking at. Based on the tree, Orsina and Rufidorsia are clearly closely related, but are still monophyletic. Based on DNA sequence data, the Sericola, Orsina, and the Rufidorsia are shown to be on different clades. They descend from recent common ancestor and its descendants. Once again, the red dots and the branches represent bootstrap values of 95% and over. Conclusions and future directions. We need to look at the ranges where Rufidorsia intermedia and Gesneriaceae are found to see whether or not they're really two species. The ranges overlap significantly and they share morphological similarities. We do not know for sure that they are different species. From our DNA sequence data and various morphological characteristics, Orsina and Rufidorsia are monophyletic. Firstly, you have a translucent pointy berry in Orsina versus a flat and white berry in Rufidorsia. You also have a reddish pink throughout the flower of Orsina versus a red dorsal top and white bottom flower in the Rufidorsia. You could lump them into one genus, but I've gone over characters which show that they are separate. The geographically isolated Orsina mexicana needs to be explored and located as it is found so far from the other members of Orsina and Rufidorsia genera. We are very interested in the recollection of these specimens. Here are acknowledgements. We are very grateful for all your support. Lastly, here's a photo of our Lawrenceville group. Thanks again, guys.
All right. All right. Good job, everyone. So that is the end of the student presentations. And so if there are any questions, I'm looking at the chat here. And so far, I got a comment from Angel Lara, VP of Horticulture. He is going to accept our challenge and produce or grow a Cobananthus with a mature fruit. So we have to figure out how to self-pollinate that and see if we can actually get some images of what those look like. So yeah, if you can ask me questions in the chat. You can raise your hand, and I'd be happy to answer any questions or forward the questions on to students. Let's see here. All right. Oh, okay. So I have a question here from Andrew Swanson about speculate about the selective pressure behind anisophilie. So yeah, that is something that we see quite often in the epiphytic genera, and I'm not really sure about the selective pressures that would result in that sort of feature. You know, when anisophilie, so if you have two pairs of leaves or two opposite leaves, and one is big and one is small, we often see that associated with epiphytism. So any of the other, anyone else have, Jeremy, Laura, you have any opinions on that? I'm not really sure on that. So I don't have a better answer for you, sorry. I'm just curious, anyone there ever see a mature fruit of Cobananthus? We're really interested in knowing if, I know a lot of you grow Cobananthus, and I'm just wondering if any of you have been able to pollinate it and get it to produce seeds. Let's see if you do, let me know. You can email me or email any of us. We've been working on this now. We've been here for a month, and so we have, I guess we came here with a lot of questions, but we ended the month with even more questions, which is, I think that's an exciting thing about doing a phylogenetics or a systematics project on biodiversity. Yeah, so there's a question, there's a comment here by Bruce Holst that anisophilie is something that occurs in a lot of flowering plant families, not just Gesneriaceae, and it's correlated with epiphytism. So the families that Bruce Holst mentioned are Melostomataceae and Solanaceae. So you can, yeah, that's for sure. You can see anisophilie in those groups, especially when they are epiphytic, when they have that habit. Let's see what else here. So great, I really appreciate it. I got a lot of positive feedback here from people who enjoyed the presentations. So thank you very much. That's really kind. Oh, so I have a question here. Where does this research go? Where does it follow up? Well, that's a great question. Yes, we would like to continue doing this research. The Central American phylogenetics work, the diastema, there's a lot of things there that need to be named. I hope that you saw that we included in parentheses the names of things that are new, or we think that are new, based on Jeremy Keen's extensive work in herbaria, looking at lots of specimens. So hopefully this week, actually this month, when we worked with the students during the day, and then at night we were actually writing up several papers. We'll probably by the end of this month, we'll publish 7 new species. So we're working on it. There's that, and who knows? Maybe maybe we'll be back at Selby next summer. You know, that would be a great thing. So let's keep our fingers crossed on that. So I have another question here. Let's see. So there's a question here from Dr. Laura Clavijo about the presence of Anopheles leaves, and she said that it's related to the dorsiventral shoots that facilitate the climbing of the epiphytic habit. So dorsiventral, it's sort of like if you think of a fern frond, you know, they don't go up and they're not horizontal. They sort of come up and they dip down. That's dorsiventral. We call that in Gesneriaceae that type of habit. And it's Dr. Clavijo suggests that this is related to optimization on light capture in the understory of forests. And a paper on that is by Givnish. Givnish is a botanist, a systematist. I think he's at the University of Madison, Wisconsin, who does work on Bromeliaceae. So if you wanted to look that up, you could probably find some of his publications on that. Here's a question. Here's something. Oh yeah, this is really interesting. This came from a Gesneriaceae Society panelist, anonymous here, but commenting that Jan Miraman provided many of the cuttings of Alsobia baronia. This is the one that is not on GenBank that we provided for the first time data from this study. That this person got this from the Belize Botanical Garden in 2010. And yes, this is true that he was told, or she was told, that the baronia grew on rocky outcrops. If you look at it online, you can see that they are lithophytes, or they grow on rocks, specifically on limestone. You get these huge like sinkholes, and I think Bruce Holst, he went down one of those sinkholes, rappelled down a sinkhole on a rope, and sort of dangled in the middle of the air and collected them. And that's true. That is true. That shows that they don't grow in other plants. They are lithophytes, so they grow in other rocks, and that would definitely be the case. So, all right. Oh, that was from Ron Meyer. Yes, Ron, thank you. And by the way, Ron Meyer has shared a lot of images. He has definitely promoted Alsobia baronia as a cutting in horticulture for a very long time. So it's nice to see that we finally have names on those. It's just from above from Mike. Okay. So I have a question here. From So I think there's a question here from Mike, but I don't see it. Can you just repost it? Oh, is that the question? So the question is the takeaway from the research. Why don't students, why don't I started off, why don't you end with any takeaways? Do you have any takeaways you want to share with our? By the way, we have over 80 people who attended, so thank you for coming. And then I think we'll end it. I think we're at 1 o'clock. So any takeaways, or Laura, Jeremy, Tori, you want to end the presentation off with any takeaways?
Well, I think the question was more like, I think it would be nice to hear the students first.
I think that
okay.
Wait, do I go?
Yeah, you go.
All right. Well, I think that in general, this research experience is very eye-opening for me, especially because I come from like more of a mathematical background. I think that just seeing the whole world of like plant biology expand and seeing how intricate and like interesting a lot of the things are. I think especially for me, one of the most eye-opening moments was just seeing like how the samples was generated and how like we use the technology, and we use a software called Genius. We're able to kind of like condense and see our data in new ways. And at least for me, that was pretty eye-opening, seeing like the intersection between technology and like basically biology. So that was pretty cool.
In addition to that, I would say like my takeaway was that it can be like hard work and it might not always make sense, but it's what you make of it. And next year I'm scheduled to take a course studying like the DNA and fruit flies. So this definitely gave me a nice intro to that and taught me valuable lessons.
Yeah, to add on to that, this project was basically an extension of what I've done with Dr. Clark about 2 years ago. So it's really interesting to keep exploring the biodiversity and of the Gesneriaceae family. Really interesting stuff. And I'm also scheduled to take the same class as Ben as next year, so that'll be pretty exciting.
Well, the other three already said a lot of what I was planning on saying, but I guess also just like all the experiences that we had here were so amazing. And I'm so glad that we had this opportunity. Just working in person in the lab, working with Dr. Keene, Dr. Clariho, and Dr. Clark, and Tori in person was really amazing as well. And also seeing the herbarium, because I've never, I don't think I've ever been to one before and seen collections like that before.
Okay, everyone, thank you very much for coming, and we are tuning out. So thank you, Selby Botanical Gardens. Thank you very much. We really appreciate your hospitality. Thank you for letting us come here. Bye.